Good morning, everybody. Thank you very much for your active participation yesterday during the talks. We hope you have enjoyed the day and also the social activities in the afternoon. And we hope you enjoy the day again. We are starting with Felix Heeger, who did his bachelor at the University of Berlin, and who is finishing his PhD also at the University of Berlin and the Leibniz-Institut of Freshwater and Ecology. And now he's working as bioinformatician at the Institute of Materials and Testing in Berlin. So in a project looking for microcolony of fungi growing in buildings. So I think it's a very interesting talk.
Well, I'm not going to talk about the buildings.
Yeah, but about fungi?
Yes. Okay, well, some of you look a bit tired. I hope the talk is exciting enough to keep you awake. As mentioned, this work was done at the Institute for Freshwater Ecology and Inland Fisheries, but it has nothing to do with fish. It's about fungi, but about aquatic fungi. And the first thing you need to know about fungi is that they are a very diverse group. So the estimation at the moment is that we might have up to 5 million species, but so far only 140,000 of them are named, and even less we have sequences for. And it's a really big group reaching all the way from the microscopic mushrooms up there in the right, and then over food molds a little bit to the left, and then to microscopic species, which are mainly what we are interested in when we are looking at freshwaters. So, quick recap on the metabarcoding. We start with a sample of fungi in our little Eppendorf cube. And then we extract their DNA and we amplify a certain marker gene, which then is sequenced via high-throughput sequencing. We end up with millions of reads, which then are processed and clustered into what we call OTUs, operational taxonomic units, which hopefully, if we did everything right and everything works well, represent species, but normally they don't. And then is the part where I'm mainly interested in, which is the classification of these OTUs. So what did we actually find? You can also do some interesting stuff with just the OTUs, like richment, how rich is your community, diversity, and so on. But if you want to do any ecological, meaningful research, you need to know what you are actually looking at here. And so for that, we need a marker gene, as mentioned before. And normally in fungi, we're looking at rRNA genes. So we have this operon of rRNA genes with the SSU on the left and the LSU, or 28 and 18S, on the right. And then in the middle between them, we have the 5.8S gene. And between these, there are the internal transcript spacers, the ITS. And the ITS1 and ITS2 and the 5.8S together are called ITS region. So if we're looking for a marker, remember yesterday's talk, we need conserved flanking regions for our primers to bind. We need a variable region in the middle where we actually can identify the species and differentiate the species. And we are actually constrained in what we can do in terms of lengths by the sequencing technology. So for fungi, the most common marker is what was suggested as a standard marker in 2012 is this ITS region. So you basically put your primers in the SSU and the LSU, so they are more conserved, and then you have the variable ITSs, which are then giving you the resolution you need. The problem with that is that this region is too long in a lot of fungi, so it has a lot of variability in the length, but in a lot of fungi it will be too long to sequence it with ITS, with Illumina. So most people actually use only one of the ITS regions. In that case, you can put your primer in the 5.8S and the other in either the LSU or the SSU, and you're fine as well. And this has a really high variability, which is good for species distinction. So I want to talk a little bit about how good these classifications are, and to do that, we need to understand a few things. The first is that each of these OTUs will be classified to a certain taxonomic level. It's not always classified all the way. So for example, the example all the way at the bottom, we have a genus classification, but then the species is unassigned, right? And if we know the actual true classification of our OTU, then we can compare that to what the algorithm gives us and define them as either correct, if they are the same, like the blue ones at the beginning, or wrong, like if they're different from the true classification at that level, which is red here, and then we might also have some level where it's unassigned. So when we look at the ITS in silico, this looks really good. So if we just take a common ITS database and classify its sequences against the same database, then we end up with something like this. We have 100% of our species, of our OTUs classified at the higher levels, and then even at the species level, it's up to 90% or even more classified, and we don't get any wrong classification. Sometimes the algorithm might say, I don't know actually what this is. It might be this species or this species, but we don't get anything wrong. So that's good. The problem is, if you do this in practice, and especially on one study we had in lakes. So these were the 20 lakes in northern Germany where we took samples in October 2014, water and sediment, and then we tried this classification with ITS, and this is what we get. I mean, this is extreme case, of course, but we have like a lot of unassigned sequences, and we have some fungi at the bottom, and some of them are also classified further, but most of it is completely unassigned. And this is really surprising, because you would think that at least if we don't know the species, then we could at least get a genus classification or family or whatever, or at least phylum, or even tell us that it's fungi. That would be nice. So why is that? My working theory at the moment is that the ITS is in fact so different, that so variable, that if you have not the very closely related species in your reference database, then it basically doesn't know where to put it at all. It's just very different from everything in the reference database, and then you get no classification. And indeed, if we test this, so we have these pictures again. At the bottom, we have the picture from before where we didn't do anything to the database, everything's fine. If we now go and remove all the sequences from the same species from the database before we classify our OTU, so we know which species it is, and we remove all the sequences from the same species, of course we do not get the correct species classification anymore. That's impossible, because there's none of this left in the database. Unfortunately, we also get some wrong classifications, and in addition, you can see that even at the higher levels, where and I actually made sure that for each of these sequences there were still from at least the same genus or the same family, there were still sequences in there, but it really breaks down, and this gets only worse if you remove species from the same genus and from the same family and so on. So what to do? The idea would be that we might use a less variable marker, which, for example, we could use the 5.8S, which is very conserved. It's, I think, even more conserved than the other two, and it's small, so we can sequence it easily in addition. And so to test this, we look again. We have the 5.8S now on the right, and we immediately see that even if we use the whole database without removing things, it doesn't really work as a species classification because it's too conserved. But the nice thing is, even if we remove data from the database, the high-level classifications stay stable. So the kingdom and phylum level classifications stay completely stable, and even the class level works quite well. So we decided that we were lucky, because when we look at our marker, so we sequenced ITS2, but in fact we had sequenced this piece, because there's a very convenient primer binding site at the left side of the 5.8S, and normally you would just throw away all the small piece of the 5.8S that you sequenced. So we used that and included that in our analysis, and this actually gave us this picture. So one of the things you will immediately see is that one thing is that the 5.8S database that we used actually also contained non-fungal sequences. So now we find that actually, I don't know, a quarter of our sequences are actually plants. So we had some off-target amplification. This is also important to remember. This is a database bias. If your database only contains fungi, you will only find fungi, and maybe you actually sequence something different. But also for the fungi, we have at least phylum. So I restricted the 5.8S to phylum level classification because I'm not sure we can trust it further than that. But this already gives us some information on the phyla. And now you might ask, but phyla, that doesn't seem very interesting because that could be like anything, right? Well, in the special case in the aquatic fungi is that really we don't know nothing about them, and we are very happy with this basic information is what I'm saying. And also the problem is much more pronounced in aquatic fungi because they are not very well represented in the databases. So you have this problem much more than otherwise. I think if you would do something similar with soil fungi or something, you might get much better results with only the ITS because the database representation is better. But I mean, we also saw in the talk yesterday that even in soil you have the same problem. Okay, so now we got a bit greedy and we thought, oh, summary, sorry. Summary, if we use the 5.8S as complementary marker, this can give us, because it's more highly conserved, we get better classification for unknown species. And this is especially interesting for us because we're looking at these basal fungal lineages, which are common in freshwater, that are not very well represented in the databases so far. And additionally, because we used non-fungal sequences in the database, we also get non-fungal classifications. Okay, so now we get a little bit greedy, and we thought, what about we sequence even more? And we came up with this piece here, which now basically contains almost the full SSU and roughly 2/3 of the LSU. And then we call it a little bit hyperbolically full operon sequencing. Now, this is 4.5 kb, so there's no way we can ever do this on Illumina or any other short-read sequencing. So we move to third-generation sequencing. And in this case, this is PacBio. We can get up to 30,000 base pairs read lengths, even more, but that's enough for us. It's reasonably high throughput, so you get, on one of their smart cells, they call them, you get around 30,000 reads. And you can run up to 8 of these in one run, and they cost you about 500 euros if you have a good deal, and yeah. The problem is with all the long read sequencing, the error rate is high. So for PacBio, it's around 40% in the raw reads. But we can reduce that when we do something that's called circular consensus sequencing. So the setup of the PacBio sequencing is that you have your initial DNA piece, and then you add the hairpin adapters at the end. And this is done by blunt end ligation. And so basically you get a single-strand DNA loop, which the polymerase will then run around, or actually the loop will be going through the polymerase. And this is where the sequencing is done, basically. But this also means that if your initial insertion was small enough, it can go around several times. So like always, the polymerase will basically just fall off at some point, right? But if your initial piece is short enough, then you will actually get multiple reads of your insert. And so I tried to picture that a little bit there, that you have like 3 passes, as they're called. And they are connected by this hairpin sequence, which you ignore. But then if you have these three, they will have random errors, and importantly, in PacBio, the error distribution is actually random. So if you align them and then you make a consensus of that, in theory, the error rate should be 14 times 14 times 14%, so very low. Of course, this in practice is not exactly how it works, but we can reduce the error rate a lot to roughly 1%. Now that we have this long piece, one of the interesting things is that you actually, if you want to do compare to a reference database, you have not one reference database anymore because they are restricted to shorter parts. So for the ITS, there's a UNITE database, especially for fungi. For the SSU, we used the Silva database, which is mainly focused on bacteria, but also contains fungi. And then for the LSU, we used the RDP ribosomal database project, LSU database, which also contains mostly fungi. There's other options, but this is what we used. And then we get a quite interesting result, which is, so this picture is a bit complicated. You have for each of the OTUs, you have 3 bars on top of each other in the 3 rows. And then the depth, or the height, represents how much they were classified. So if it goes all the way to the top, we have a species level classification. One down would be genus, family, and so on. And they are colored by the phylum. So what we see here is that for the ITS, we have basically the same problem again. That for a lot of the species, we don't get any classification at all. It works quite well for the orange and the red, which is the Ascomycota and Basidiomycota. These are the more well-studied species. And if we get classifications, they are normally very good. So they're almost always to the species level. But then for the ones that are actually less well-studied, like the Chytridiomycota and Cryptomycota, and these are very important in freshwater, we get nothing. But then we have the SSU and the LSU, and especially the SSU gives us much better classifications at lower or at less specific taxonomic ranks, but this is still interesting for us. And the additional benefit of this is that now that we have these combined data, we can actually see a synergy effect between the databases. So we have a sequence that is, let's say, classified to the order level by the SSU, and we don't get anything from the ITS. So now, in theory, we could go and put this sequence, this ITS sequence that we have, and we know that it's from the same species because it came from the same molecule. We can put it into the ITS database and at least have an order level classification. So the next time we are running with short reads, this will be a huge benefit. And yeah, I think then that's it, summary. So high variability markers can be used in combination with low variability markers. And this will give us a high resolution by the high variability markers. And it will also give us a broader classification coverage by the lower variability markers. And this is especially important if the reference databases are incomplete, for example, in the case of basal fungal lineages. And yeah, the more long reads we have, the more we can sequence, so these approaches become more viable with new sequencing technologies. Yeah, thanks to my collaborators, especially Elizabeth Bourne, who did most of the lab work, Michael Monaghan and Christian Wutzbacher, who were very important in coming up with these ideas, and then the DSMZ, who did the sequencing, and the Leibniz-Institut Association, who actually funded this research. And you for your attention.
Any question?
What did you use for the taxonomic assignment in the first place when you were doing the ideas versus?
It's a last common ancestor approach. So we blast first, and then we select some of the best matches, and then we get the last common ancestor. So go up the taxonomic tree basically to the one where we can say, oh, we don't know if it's this species or this species, but it's definitely in this genus.
I'm just wondering about costs. So with only 30,000 reads, and let's say you want 30,000 reads per sample.
Yeah, reasonable.
And you want to do 100 samples or something, does it start to get astronomical?
Yes, I think so. I mean, we only did this on, I think, 6 samples, just to prove that it works. I think it will not be the standard method that you use if you're just, if you're just interested in the ecological applications. But it might be useful if you're looking at something very new, something that has not been studied before, to get a ground level and improve your database, and then maybe use Illumina sequencing later to map to the reference.
Or even to add new taxons to the databases.
Right, and I mean, this is like in aquatic fungi we have the Cryptomycota, which is a phylum that was introduced, I don't know, 10 years ago maybe, and we know like 3 species in it. Yeah.
Isn't any trend that look like just a bacterial database but still found a lot of?
Oh, yeah, it is mainly bacterial, but it's very big and it also contains fungi. They started it basically as a bacterial database, and it's like, I think, almost 2 million sequences, but there's also some fungi in there.
Okay, so it's not that you get everything.
Are you thinking also, or were you thinking of building custom databases, specifically for these? I'm thinking, like we talked yesterday a bit, about the local variation between the lakes, for instance. And it's like, I'm wondering if you're trying.
We haven't done anything in that direction yet, but I think it would be definitely very interesting, especially because if you have this, then this also means, because a lot of the fungi studies are using different markers, right? I said the ITS is the standard, but also people are using a lot of other things, and especially for certain groups like the Glomeromycota, which are the symbionts of plants, the root symbionts, they're using a lot of LSU there, and then if we have actually a database which contains the full operon, then we can compare these to each other. We would find the same reference sequences there. So I think that would be definitely a very interesting project.
So do you think you can find the same amount of Glomeromycota using this approach and using the MarkZam database, the specific weighter?
I don't know, because this is like for the aquatic fungi.
I didn't see if you have Glomeromycotas.
Yeah, I think there was like one OTU that was that, but mainly they are not very important in the water. So yeah, I think then somebody else would have to do this for soil.
Yeah.
But there is a paper, I think, where they use not the same length, but a little bit shorter for Fusarium or something, and this, I don't know, they use a primer set which is also often used for nested PCR in Glomeromycota, where they have first a big piece and then they sequence a smaller, amplify a smaller piece for sequencing. So yeah, it's definitely applicable.
Okay, thank you. Thank you.